Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about short distance signaling in the body. So you'll recall from our previous lesson that cell signaling, cells communicating with each other in the body, is almost always going to involve some kind of signaling molecule, whether it's a hormone or neurotransmitter or something else. A signaling molecule is going to travel from the cell that secretes that molecule towards a target cell, and that ligand is going to bind to a receptor that is inherent to or possessed by the target cell. Now, we're going to talk about all different types of signaling in the body over the next few videos, but what you're going to see is what actually separates one kind of signaling from another is, number one, how far the ligand has to travel from where it starts to where it's going to end up, and number two, through which fluid medium it's going to have to travel to get there. Now, you can relate this to how you travel in your everyday life. What mode of transportation you choose to use is going to very much depend on how far you have to travel. Are you going down the street? Well, you're certainly probably not going to have to take a car or an airplane to get there. Are you traveling to another country? Well, you're probably not going to want to walk or ride your bike to get there. So you're going to see that cell-to-cell communication in the body uses the same sort of logic. The way in which molecules travel from place to place is going to very much depend on how far they have to go to get there. Now, as far as long-distance communication goes, we are going to have full lessons, full units, full series of videos dedicated to the two major types of long-distance communication in the body, that being neural communication and hormonal or endocrine communication. So we're not really going to mention them very much or even talk about them very much in this particular video, but rest assured we will have plenty to say about them in the future. So back to local communication. What we mean by short distance signaling or local communication is that the cells that are communicating with each other are located very close by to one another. So usually that's going to mean that these cells are a part of the same tissue, and we are not going to have to use nerves or the bloodstream in order to get the message delivered from where it starts to where it ends up. Now, to that end, there are several different types of short-distance communication. The first we'll mention is called paracrine signaling. In this illustration, we have two cells here. The cell on the left is the one that is going to send the message, and the cell on the right is the one that is going to receive the message. So you can see the blood supply up here, but what you're going to notice about paracrine signaling is that the signaling molecule has no need to enter the bloodstream at all. So paracrine signaling is going to utilize the interstitial fluid as the transport medium. What you'll notice here is that the cell that is doing the signaling synthesizes and secretes the ligand, and that ligand diffuses through the interstitial fluid until it finds a receptor on a target cell. And then, of course, as we've said before, the engagement between the ligand and the receptor is going to produce for us our response. Now, some specific examples of paracrine signaling is blood clotting, inflammation, tissue repair after injury. Neurotransmission is actually kind of a special application of paracrine signaling, even though it's not strictly local communication. But the basic idea here is that the only way using the interstitial fluid as your transport medium would work is if it's local communication. The interstitial fluid does not have a flow to it like the bloodstream does. You can't deposit a ligand into the interstitial fluid and then count on that ligand to be carried far and wide. There's no flow there, okay? It's like still waters. So what you're counting on here is the ligand reaching its destination by random diffusion, or Brownian motion as it's sometimes called. So you can't really count on the ligand traveling very far in a short amount of time if you're using the interstitial fluid as your transport medium. And for that reason, this really only works for short distance communication. Now, another type of signaling, which is almost identical, is called autocrine signaling. This employs the exact same principles, except in autocrine signaling, the cell that sends the signal and the cell that receives the signal are one and the same. We're not going to talk about autocrine signaling in very many instances, but one specific application of this is cells will sometimes release a small amount of signaling molecule that then binds to a receptor on the self-made cell, and then that activates some sort of positive feedback loop that causes the cell to start producing more and more signal, and that can actually cause an increase in interstitial fluid concentration of the ligand, and that could potentially make paracrine signaling a little quicker and a little more efficient because you've actually kind of gotten yourself to produce a larger amount of signal. So to give you a couple of examples of paracrine signals, some of which you may have heard of, some of which you may not have heard of, histamine you may have heard of. You're probably more familiar with antihistamines than you are with histamine itself. Histamine is a small molecule that is often responsible for allergic reactions. It affects not only the endothelial cells and the epithelial cells, but also smooth muscle cells to cause redness and inflammation due to increased blood flow, the central nervous system. So antihistamines, as the name suggests, are medications that are meant to limit the histamine response on target cells. Cytokines are a very diverse group of signaling molecules that affect many, many different cells in the body, and oftentimes they function to activate the immune system in one form or another. Nitric oxide, which we'll talk a little bit more in depth here in just a minute, is a small molecule that affects smooth muscle, and more specifically affects smooth muscle contraction in vascular settings, helping to regulate blood flow. So we'll kind of see a little bit more of that here in just a second. We've talked about prostaglandins before, which help to mediate inflammation. Thromboxanes and leukotrienes are very similar in that they are derived from lipid compounds, specifically fatty acids, and they have a number of different effects. For example, thromboxanes help to affect blood clotting. Leukotrienes affect the lungs and are oftentimes a big part of causing respiratory diseases like asthma. Okay, so I'd mentioned we're going to talk a little bit more about nitric oxide. This is just to give you an idea of a specific physiological process for which paracrine signaling is important. Now, in the future, when we get to our cardiovascular unit, we are going to take a look at this picture again to get an idea of the anatomy of a blood vessel. But arteries and arterioles, which are the major blood vessels that carry blood away from the heart, tend to have three different layers to them. They have an outer adventitia or externa layer that basically consists of connective tissue that helps to kind of anchor the blood vessel down so it doesn't squirt around all over the place. A middle layer of vascular smooth muscle, and then an inner intima layer of endothelial cells, which basically makes up the inner wall of the blood vessel. And then, of course, on the innermost, you have the hollow lumen, which is where the actual blood is contained. So the one we want to focus on here is the innermost vascular smooth muscle layer. So the idea here is that blood flow through big vessels like this has to be regulated through a balance of what we call vasodilation and vasoconstriction. True to what the names mean, and you probably kind of already picked up on this, blood vessels can regulate their diameter. Vasodilation opens up the diameter of the blood vessel and allows blood to flow much more freely. Vasoconstriction restricts the diameter of the blood vessel, meaning that it has a much smaller lumen, meaning that blood will not flow through nearly as well. This is going to be something that we talk a lot about when we start talking about the autonomic nervous system and then into the cardiovascular unit. But basically, for now, you can appreciate that the contraction and relaxation of these smooth muscle fibers that are in that middle smooth muscle layer help to regulate blood flowing through particular arteries and arterioles in the body. So if we look at the kind of anatomical relationship or spatial relationship between the innermost endothelial cells that you see here. So if you're kind of picturing this, the blood plasma would be up here. There's your innermost endothelial cell layer, and then here's your smooth muscle fibers down here. As I was saying, it's the contraction and relaxation of these vascular smooth muscle cells, or VSMCs, down here that helps to determine the diameter of the blood vessel and thus the rate of blood flow. So I mentioned that paracrine signaling has something to do with this. So if you look at what's happening up here in the endothelial cell, we need to focus on an enzyme called nitric oxide synthase, eNOS for short, for endothelial nitric oxide synthase. This is an enzyme that has two substrates. One of them is familiar to us. This is arginine, which is one of our 20 amino acids, and then oxygen, which is of course delivered from the bloodstream. This enzyme, eNOS, will combine these to form nitric oxide, which is a small gas. So nitric oxide here, as a small nonpolar gas, can freely diffuse out of the endothelial cell and into the interstitial fluid, and then across the membrane of the vascular smooth muscle cell. And we're not going to get into the specific details here, but nitric oxide will start to affect the internal structure of the vascular smooth muscle cell and cause it to relax. When the vascular smooth muscle cell relaxes and all of the vascular smooth muscle cells in the area start to relax, the overall effect we get is vasodilation, which will help to improve the blood flow. Now, if you're interested in learning more about this, you might check out the mechanism of action of caffeine. Caffeine is a known vasodilator, and I think when you do some research, you'll find that caffeine specifically affects nitric oxide production. Okay, so to finish up this discussion on local communication, let's talk about gap junction signaling. We've mentioned gap junctions before as being a major junction protein that are found in the intercalated discs found in between cardiac muscle fibers and cardiac muscle tissue. So as a reminder, gap junctions connect the cytoplasm of adjacent cells in tissues like cardiac and smooth muscle so that molecules that are small enough to flow through these open gap junction channels can allow those molecules to move from the cytoplasm of one cell to the cytoplasm of another, as long as those cells are connected like that. So this type of gap junction signaling is going to be especially important for cells that are excitable, for which cardiac muscle and smooth muscle fibers are excitable. The reason why this is important is that charged ions like sodium and calcium can pass through gap junctions. They are small enough to move through open gap junctions. So look at this scenario here. This cardiac muscle cell right here has opened a sodium channel. This is causing sodium to flow into this cell from high concentration in the extracellular fluid to low concentration in the intracellular fluid. And as long as this sodium channel stays open, this cell is eventually going to fill up with positively charged sodium. This is going to greatly affect the membrane potential for this cell, and this cell is going to get excited enough to produce a contraction, okay? So what you'll notice, though, is that the cell on the far left has gotten excited because it's got all this sodium inside it now, but as of right now, the other three cells are not excited. So this would cause this cell on the far left to produce a contraction, but these three cells to the right of it are going to stay relaxed because they are not excited yet. This is kind of a problem because as far as cardiac muscle goes, as far as the function of your heart goes, we want big sections of what we call myocardium, which are cardiac muscle fibers connected by gap junctions. We want them to all get excited at the same time and all contract at the same time. That's going to be the only way we get big, strong enough contractions to pressurize blood and pump it out to the body. So we need to figure out a way to get all of these cells excited at the same time so that they can contract and relax at the same time. That is why the presence of the gap junctions is so important. If one cell becomes excited, the sodium ions can diffuse through these open gap junctions and get all of the cells excited at the same time so that they can all contract at the same time. So we'll have a lot more to say about this when we get to our section or our unit on the cardiovascular system, but this is a good thing to introduce for right now. Okay, that's going to pretty much do it for this video. Here is a list of vocabulary terms you might want to be aware of. And as far as checking your understanding goes, number one, what do long distance and short distance signaling have in common with each other at the molecular level? Number two, what function do gap junctions serve in the beating of the heart? And number three, how could injuring yourself cause local inflammation through local signaling? Okay, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section. I will see you next time.